Good wishes to everyone. Today we will discuss about the chromosome structure and functions. So, what's a chromosome? As you know, the chromosome is indicated by the divided cells like this and the thread-like structure with different colors. That chroma means it's a color. Then usually the chromosome was first found in 1875, first discovered the thread-like structures by E. Strasburger, which appeared during the cell division. And these thread-like structures were called as the chromosomes due to their affinity for basic dyes. And the term chromosome is derived from the Greek words chrome means color and soma means body. So this term was first used by Waller in 1888. The year in which it is found is 1875. And chromosomes contributed to the division of the cells and they are prime importance as they carry the genes which are the hereditary material. Then as usual, the chroma means it's a color. I would say, or in simple terms, what you can say, it's a colored material in the body. It's a thread-like self-replicating genetic material which is present inside the cell, and especially it's present in the deoxyribose nucleic acid and package found in the nucleus of the cell. So where the chromosomes are found, they are all found in the nucleus of the cell. And chromosomes are soon dividing during metaphase stage of the cell division. And when the cells are stained with suitable basic dye and viewed under the light microscope itself, we can find the chromosomes. That is the specialty of these chromosomes. So all types of the higher organisms, the eukaryotic cells or the animals, they have the chromosomes, the very well organized nucleus containing the definite number of chromosomes, and it is very characteristic to each species. It's a fixed thing and it is defined and shaped. So the Waldeyer coined the term chromosomes. Then the term chromosome number is the number of chromosomes found in the somatic cell and represented by 2n, or it's also called as the diploid, means double number. The gametic chromosomes number is half of the somatic chromosomes number and represented by n. It's also called as the haploid cells. And two basic copies of the chromosomes are usually identical in morphology, gene content, and gene order, and they are known as the homologous chromosomes. And the chromosome number is a constant or fixed in a defined species. All the members of the species ordinarily have the definite and generally the constant somatic and gametic chromosomes. The somatic chromosomes number is found in the somatic cells of the species and the represented by 2n, or it is diploid, whereas generally the somatic cells do contain two copies, means soma means body, body cells do contain the chromosomes of two sets, whereas the gametic cells or the sperms in a male and the ova in a female do contain the haploid number or half of the number. So the if both copies of the chromosomes are looking identical and other thing, they are called as the homologous chromosomes. And the gametic chromosomes number is exactly half of the somatic chromosomes and is represented by n, means it denotes the number of the chromosomes found in gametes of the species. So the chromosomes are two types. One is autosomes, that is the control characteristics other than sex characters or carry genes for somatic characters, body characters only. It is not going to determine the sex of any individual organisms. So the chromosomes are involved in sex determination And humans and most of the mammals do have two sex chromosomes. It is noted by X and Y. It is different in different species. So they are also called as the heterosomes. And females have X chromosomes in diploid cells. Males have X and Y chromosomes. In birds, the female ZW is heterogametic and male is the ZZ is homogametic. So this is the chromosome number photo. Haploid is usually the single with either X or Y. The diploid is 2N. It is either XX or YY. So triploid, it is containing the three pairs, and tetraploid, it is four planes. So all these things are different species. So what's the size of a chromosome? It shows a remarkable variation depending upon the stage of cell division. Longest and thinnest during interphase, then hence it is not visible under light microscope, but smallest and thicker during the mitotic phase. And chromosome size is not proportional to the number of genes present in the chromosome. And the chromosome morphology, the outer covering of sheath of the chromosome is known as pellicle, which encloses the matrix. Then within the matrix, it lies the chromatin. Then Alexander Fleming, introduced the term chromatin in 1879, and it changes during the cell division. And in mitotic metaphase, the chromosomes, the following structural features can be seen. Number one, the chromatid, the centrioles, telomere, then secondary construction, chromomere, then the chromonema and matrix. Oh, so we will see them individually. The chromatid is having each metaphor's chromosome appears to be longitudinally divided into two identical parts, each of which is called as the chromatids. Then chromatids of the chromosomes appear to be joined together at a point called as the centromere. This is the point where the chromosomes threads are going to join, and the both of these are called as the sister chromatids. Then the chromatids of the homologous chromosomes are known as non-sister chromosomes. Very similar to the other thing. There is a difference between the centromeres. Then it is also called as the kinetochore, which is a protein in structure. Then they are called as the sister chromatids. What is a centromere? It is the region where the sister chromatids appear to be joined during the metaphase, and it's also called as the centromere. So it is centrally appears the constriction and called as the primary constriction. And this primary constriction, it helps in movement of the chromosomes to opposite poles during the anaphase of the cell division. As you know very well that the cell division contains anaphase, metaphase, telophase, etc. So the chromosomes consists of two disc-shaped bodies called as the kinetochores. And normally chromosomes are monocentric, having only one center, except certain type of the cells. So the depending upon the position of the chromosomes, we can classify them as the metacentric, where the chromosome is located exactly at the center of the chromosome. So these such chromosomes assumes V shape at the face, whereas the submetacentric is located, the center is located at one side of the center. That arm V is longer than that of the other two arms. Acrocentric is located close to one end at the chromosome, and thus giving a very short and a very long arm. And these chromosomes acquire J shape or rod shape during the anaphase, and we will see the photos. And telocentric, it is located at the one end of the chromosome, so that the chromosomes has only one arm, and these chromosomes are called as the eye shape or rod shape. So here you can see that the telomere are called as, so the two ends of the chromosomes are called as the telomeres, either ends. So they are highly stable and not fused to unite telomeres to other chromosomes due to the polarity. They do have the different polarity effect, so they repel each other. Any broken end of the chromosome is unstable and can join with a piece of any other chromosome. But the telomere impart stability to the chromosome, which retains its identity entire cell cycle and for many cell generations. So this is called as secondary constriction. So the constriction is narrow region other than that of the center is called as the secondary constriction. The chromosomes having the secondary constrictions are known as the satellite chromosomes. Here they are called as the satellite chromosomes. And chromosome may possess secondary constriction is one or both arms of it. So the chromosome end distal to the secondary constriction is known as the satellite. And this is defected by, this is called as the satellite of the chromosome, and production of the chromosome is associated with secondary constriction, and therefore it's also called as nucleolus organizing region. So the region is called as the nucleolus organizing region, and satellite chromosomes are often referred to as nucleolus organizing chromosomes. Then chromomere is, this is the structure of a chromomere. It is some species like the maize, rye, etc. So the chromosomes in the parenchymatic region, stage meiosis show small bead-like structure called as the chromomeres. So the distribution of chromomere in chromosomes is highly characteristic and it's also a constant pattern. The pattern distribution is being different for different chromosomes and they are clearly visible as dark staining bonds in the joint salivary chromosomes. So these chromosomes are tightly folded with the DNA. So here you can see that this is containing chromomeres, a strip-like structures, chromonema, then the pellicle and also the matrix, and there will be a primary constriction. And this is the structure of a chromosome with chromonema, means the chromosome consists of two chromatids and each chromatid consists of thread-like coiled structure called as the chromonema. These are all the chromonema. The term chromonema was coined by the Vedovsky, a Russian people, in 1912, and the nemata form gene-bearing portions of the chromosome. So this is the gene-carrying structure of the chromosomes, and this is the satellite body, and this is nick is called as secondary constriction. Whereas the chromosomes matrix, the it's also the one of the thing, the mass of the achromatic material which surrounds the nemata is called as the matrix, which is surrounding all these things. And matrix is enclosed in a sheet, which is known as the pellicle. And both matrix and pellicle are non-genetic material and are appear only at the metaphase when nucleolus disappears. Later, they are going to be not there on the chromosome in the regular pattern. And the composition of the chromosome, it is consists of a main material called as the chromatin, and Fleming introduced the chromatin word in 1879. Then there are two other structures like the acetocarmine and fulgen, especially in case of the chromatin, it is classified. The darkly stained regions are called as heterochromatin. Then, while lightly stained regions are called as the euchromatin. And this differentiating staining structure is also called as the hetero, means heteropignosis. And heterochromatin is further classified into once again two groups. Then the constitutive, it's present in all cells at identical positions and both the homologous chromosomes of a pair. Then facultative, it varies in state and different cell types at different stages of the sometimes from homologous to the other thing. So here you can see the structure of a chromosome. Usually the end structure is called as the telomers. Then these orange color regions indicate the heterochromatin. The center one is known as the chromocenter, centromere. Then the arm-like structure is called as euchromatin. So the nuclei consists of chromatin strands as tightly packed heterochromatin or loosely packed euchromatin. Here you can see that it's heterochromatin and it's also the euchromatin. So usually the chromosomes are having the karyotype and ideogram. So characteristic feature by the set of chromosomes of species is identified. So a species can be identified by the structure of the chromosomes and number very easily. Hence, generally karyotype is represented by arranging the chromosomes in descending order of the size, keeping their centromeres in the same line. And the karyotype of a species can be represented diagrammatically showing all morphological characteristics. So such diagram are called as the ideograms or ideotypes. Then what is the special types of the chromosomes? There are varieties of the chromosomes are there. So other than the one normal chromosomes, they are called as the special type of the chromosomes. Number one, the giant chromosomes or polytene chromosomes are first discovered by E.G. Balbini in 1882 and in the salivary glands of the dipteran and hence commonly known as salivary gland chromosomes. And these chromosomes replicate repeatedly, but the daughter chromatids do not separate from each other and the cell also do not divide here. So it results in the formation of the many stranded, very giant chromosomes known as polytene chromosome and the condition is known as polyteny. So the size of these giant chromosomes is 200 times than that of the normal somatic chromosomes and very very thick. Hence they are known as the giant chromosomes. And here you can see the structure of a giant chromosome which is containing so many strands. Here you can see the centromere etc. Even though it's one, multiple strands have been joined together. So here you can see that it's also called as the chromosomal puff and a dark band and interband etc. are seen in this special type of the chromosome. And the special types of the chromosome, it is called as the lamp brush chromosomes and these were first observed by W. Fleming in 1882 and later they are detailed in oocytes of the shark fish by the Rouquet in 1892. So they are all the special type of the chromosome and these are called as the special type of the chromosome. So when they are stained looking like this and there is a area which is magnified, then we can find the special type of the chromosomes. And the third type of the chromosomes, special accessory chromosomes in many species, some chromosomes are found addition to the normal somatic chromosomes, and these extra chromosomes are called as accessory chromosomes or the B chromosomes or supernumerary chromosomes. And these chromosomes are also called as the special type of the chromosomes. Then the these are all the special type of the chromosomes in which they take a special staining and the another special type of the chromosome is isochromosomes. As an isochrome it is the one in which two arms are identical with each other gene content and also the morphology. And such chromosome is an S-sense or a reverse duplication with extromere pairing two arms. Every isomer is metacentric. Then these are all called as these are the normal chromosomes. Then these are isochrome formation is there with two short arms and sometimes it's two long arms. So the special type of the chromosomes continued as allosomes or sex chromosomes. And these are as usual X and Y structure is always there in most of the structures, wherein which the females have was double X structure and the males always they have X and Y chromosomes inside the cell, whereas they always do contain the X, Y in the gametic, whereas in case of XY, man, and XX in case of females. So they are classified according to the centromere center, acrocentric, then metacentric means the centromere is at the one end of the chromosome, that is the centromere is median. Then centromere is localized approximately midway between each, and thereby two arms are roughly each equal in. So these take a V shape along with the anaphase. So here you can see the chromatids, especially they pull their arms, and the metacentric chromosomes do have the central chromosomic end. And submetacentric, it is the centromere is located in one side of the central part of the chromosome. Then centromere is submedian, giving one longer and one longer shorter chromosome. And submetacentric chromosome may be J or L shape. Here you can see that they are J or L shape or inverted L shape also. Then acrocentric, it is the center is located to the close to the end of the chromosomes. So that's called as acro. In acrocentric refers to the Greek peak, or the P short arm is short and hard to observe. It's very tiny structure, but still it is present. And acrocentric chromosomes may be rod shaped during the anaphase of the cell division. Then telocentric, it is the chromosome based on one end of the chromosome at terminal lies at the end, and telocentric chromosome may be the large one. And here you can see it is located in the extreme end of the chromosome. So, according to the number of chromosomes, the eukaryotic chromosomes may be eccentric without any centromere, then monocentric with one center, dicentric with two centromere, then polycentric with more than two centromeres. Then how large and sequential DNA can fit into a small chromosome? Yes, now we know that the DNA is a genetic material that is carried from one species to another species through the sex cells, maybe the sperms or the ova in case of females. So what is the size of a DNA and what is the size of a chromosome? So they contain the chromatin, nucleosomes, DNA binding, and DNA. So these are all the the structures of a chromosome. So deoxyribonucleic acid with the DNA binding protein, and the nucleosomes are present, and ultimately they are present on the chromatin. And here you can see the chromosome packing. So short reason of the DNA double helix structure or Watson-Crick model, you can see this one, number one, whereas the beads on starting of the chromatin is shown here, and 30 nanometer chromatin fiber are packed nucleosome. Then section of the chromosome in extended pattern. So entire mitotic chromosome can be seen here, and it is about 400 nanometer. means it is a long structure, whereas the short chain region of the DNA helix is 2 nanometer. And here you can see that the second one is never 11, whereas the section of chromosomes in an extended form it's 300 nanometer. So histone, it can be grouped into five groups: H1, H5, H2A, H2B, H3, and H4. And they are classified into two super classes as follow: H2A, H2B, H2, H3, and H4. And the linker histones are also called as the H1 and H5. So the linker DNA is double-stranded DNA between the two nucleosome cores, that is association with the histone. H1 body, here you can see the histones are the one. Then nucleosomes cores in association with the histone, it holds the cores together. So cores of these chromosomes are held together by histones. Then nucleosome, a basic unit of the DNA packing eukaryotes in most of the eukaryotes, apart from that of the prokaryotes. It consists of the segment of DNA wound around histone protein core and fundamental repeating unit of the eukaryotic chromatin. And the nucleosome or particle consists of approximately 147 base pairs of the DNA, which are wrapped in 1.67 left-handed superhelical turns around a histone octamer consisting of two copies each of the core histones like this. So, this is the packed structure of the core proteins, especially the linker DNA and the non-condensed chromosomes without the linker histones resemble beads of string of the DNA. Like this, it will be thickly coiled inside the cell. And the DNA protein structure is stabilized by attachment to a non-histone protein. And contrast to most eukaryotic cells, mature sperm cells largely use proteomins to the package of the DNA most likely to achieve an higher packaging ratio. Histone equivalent and simplified chromatin structures have been also found in archaea, providing that chromosomes are not the only organelles that use the nucleosomes. So, apart from the chromosomes, they may be present somewhere else. And this is the structure of that particular uh, structure. Usually the DNA is present in the isolated patches. Then the active chromosomes, then during the interphase they will be there, and during the development of the cell or division, all these structures are being formed in different stages. So heterochromatin is tightly packed chromosome, intensely stained, and consists of genetically inactive satellite consequences. Then both centromeres and telomeres are heterochromatic, means stained with different stains, and you can see this particular structure in this particular photograph. And euchromatin is the one wherein which the DNA and RNA proteins are packed to form the chromatin that is rich in gene concentration, often under active transcription. Unlike the heterochromatin, it is found in both the cells with nuclei and cells without the nuclei. Prokaryotic cells also it is seen, and most active portion of the genome within the cells. This euchromatin is most active. Then the different species of the animals will have a different and fixed number of the chromosomes. So the human it's the diploid number in the soma or the body cells, it is 46. Then coming to the plants, it is 14. Then cat, it is 38. Whereas the river buffalo, what we have got, the river buffalo, the chromosome number is 50. Whereas the swamp buffaloes, it is 48. And cattle, it is 60. So there is a difference between the cattle and the buffalo chromosomes. Dog, it is 78. Donkey, it is 62. Then horse, it is 64. So whenever there is a chromosome number is different, the cross cannot happen. Whereas the goat, it is 60. Then the pig, it is 38, and camel, it is 74, rabbit, it is 44, sheep, it is 54, goat, it is 60, whereas sheep, it is 54. The mules have got 63, whereas horses do have 64 number of the chromosomes. Then gorilla is also having 48 number of the chromosomes, whereas the humans have got 46, hence the crossing cannot happen. Whereas the hamsters do have 44. Bison, it is 60, equivalent to that of the cattle, but the crossing cannot happen between a bison and uh, that of the cattle, because there is a, even though there is a number is same, but the totally the genetic structure is different. Chimpanzee, it is 48, and deer, it is 68, elephant 56, tiger 38, honey bee maybe sometimes it is 32, and male and female there is a variation. Then horsefly 12, then fruit fly it's 8, then the alligator, it is 32 number of the chromosomes are present in every cell. And Japanese quail, it is 78. Chicken and Japanese quails do have the same number of the chromosomes. And the Muscovy duck, it is 80. So like this, it goes on like the guinea pig, 64, hare, 48, lion, it is 38. Tiger and lion do have the same number of the chromosomes, but the crossing is not possible. Mouse, it is 40, musk ox 48, rat, it is 42. Pigeon, it is once again it is 80, just like the musk duck. Then pheasants do have 82, turkey 80, domestic duck 80, emu, it is 80, and goose. Most of these big birds do have the same type of the chromosomes. So that's about the chromosomes, and we will have the questionnaires later. And have a nice day. Goodbye.